to this first, welcome everyone to this first session of Virtual Evolution. My name is Mark McPeak and I'm the moderator of the session. And we'll begin with a talk from Ellen Martin. Ellen, take it away.
Hi everyone. Good morning, I think, to the Western Hemisphere, and good evening if you're in my part of the world in Australia here. My name is Ellen Martin. As I say, it's my pleasure to be able to share with you some of the early results from my PhD project here at the University of Adelaide with such a cool conference. I've been doing this project under the guidance of my supervisors, Emma Sherratt and Rachel Norris. And primarily in my PhD, I've been examining locomotor adaptations in the limb bones of birds and mammals, both to see how different adaptations to functions such as running or digging or grasping present in these groups, but also to see how specializing for these functions could impact these animals' potential to evolve for other functions. Let's see, go slide. So before I start, I'd like to acknowledge the traditional owners and knowledge holders of the land that I'm living and studying on, who are the Kaurna people, whose presence and cultural traditions are just as important to this region today as they were prior to the colonization of Australia. So I'd like to speak today about the part of my project that involves birds. Although my results are extremely preliminary at this stage, I'm excited to see what's going to come of all of it in the end. So it mainly builds on a small project that I did in undergrad that tested for the presence of this correlation between foot proportions and ecological preference in Australian birds. Namely, it's this apparent trade-off between the lengths of the toe bones, where birds that walk or run a lot have a long proximal phalanx and a very short distal one, as well as a short hind toe, which you can sort of see on the left side of this diagram where the penguin is. And birds that use their feet for grasping and gripping and climbing have the opposite arrangement, which is shown on the right. And then in the middle, you sort of get birds that walk and grasp that have this intermediate morphology. This pattern has been identified previously in North and South America, but I was curious to see if it would also present in the Australian avifauna. So using digital calipers, I took the measurements that are pictured here from a good number of species from the South Australian Museum down here. And I used this data to create the phylomorphospace figure that you can see on the screen. So this is a visualization of the main shape variation across my sample species with their phylogenetic relationships overlaid on top of it. So as you can see, the pattern that I described holds fairly true in Australian birds, with the warmer colored grasping species falling mainly on the right hand of that phylomorphospace, and the walking, wading, and swimming birds in cool colors on the left-hand side. This was particularly true in birds with anisodactyl foot shapes, which are the ones that have 3 forward-facing toes and 1 reverse toe, like they are shown on the side of the figure there, as opposed to zygodactyl birds who have an X-shaped foot and mostly tended towards the middle of the morphospace with this intermediate morphology and ecotype. This finding, as well as a few unique Australian exceptions to it that are discussed in the paper associated with this figure, got me wondering what else we might learn about bird micro and macroevolution from their feet. So my first question was to wonder whether this trade-off in foot morphology or the differences in toe configuration could impact the evolutionary potential of different groups of birds. To investigate this, I selected 2 anisodactyl species whose foot morphology reflects both ends of the trade-off, a walker, the stubble quail, and a perching bird, the noisy miner, as well as 2 zygodactyl birds with convergent foot morphologies, the crimson rosella and the northern flicker, and repeated my previous caliper measurements with a few extra widths and lengths included. My aim was to perform an evaluation of morphological integration. So morphological integration is a concept that you might be familiar with, wherein we take the covariation of morphological traits in a large sample size. In this case, I measured the foot proportions of about 50 birds per species and identify whether these measures change in tandem with one another or vary independently. If they covary tightly, which is to say if the traits are highly integrated, this could indicate that there's some factor operating which maintains foot proportions to an exact degree. This factor could be a strong selective pressure that favors such proportions, or it could be a developmental factor that tightly binds these traits together, such as genetic linkage. Either way, high integration across a structure can make its individual parts vary less independently and less independently adaptable to change in the environment, like this. However, in the other example, proportions of the toes are less integrated and change freely without relation to one another. While this may allow a structure more freedom to change shape into a new form, it may also hinder how effectively an organism can coordinate such changes across the rest of its body. I hypothesized that the quail, a basal bird with the ancestral foot form, could provide a baseline level of morphological integration, and that zygodactyl birds, which use their feet for grasping, might have an increased amount of integration between their toes to coordinate the more complex movements and positions required for their lifestyle. Similarly, Perching birds represent a return to the ancestral state of the foot, but maintain a grasping ability. Might their integration level then remain higher if their foot is used for a different purpose than the quail? Of note is that perching birds represent approximately 60% of avian diversity, and it has been speculated by a few people in the past that the streamlined and generalized nature of their foot may have played a role in their success. It's possible that a difference in the ability of their feet to adapt could have influenced their wide radiation of species. So there are several methods of evaluating morphological integration, but in this case, I use the comparison of the relative eigenvalue indices of foot length data for the 4 species. This is a measure of covariation between traits that can be implemented using the function integration.vero in geomorph package in R. So the results of that test are shown here. The highest amount of integration in the foot is indeed appearing in the rosella, however, the lowest appears in the minor, and the woodpecker and the quail fall intermediate. Although these results could imply the proportions of the foot and the rosella covary in tandem more tightly, possibly as it possesses the most complex foot behavior, the integration values for each species are not statistically significantly different from one another. But this doesn't mean that differences in the covariation don't exist on other levels. So after I'd finished looking at the overall integration of the traits in the entire foot, another approach that I took was in evaluating the covariation of traits on a more specific scale, in this case by looking at the potential modularity of the toes. In the context of morphological integration, a module refers to a structure in the body, for example, a limb or an organ, or in this case, toes, whose constituent traits covary more closely with one another than they do with the traits of a different module. This enables these modules, the functional units of the body, to be more capable of evolving relatively independently of other body parts. Here, I considered whether the forward and reverse toes, digits 3 and 1, may act as the same or as separate modules depending on how each species uses their feet. I hypothesized that the covariation between the toes was likely more important in strongly grasping species, as they were more likely to be a joint module in those cases. I tested this using a 2-block partial least squares analysis, which is a process that quantifies the amount of covariation between 2 different blocks of variables. As you can see from the scatter plots of that 2-block partial least squares test, some differences in covariation are indeed present in the 4 species. So the perching minor up in the top right, there's much less variation in the proportion within the toes, which is indicated by the smaller spread of points, but the low RPLS score and the non-significant p-value indicate that the forward and reverse modules don't covary tightly together. This could indicate that although the 2 toes are relatively independent despite their grasping nature, it is important that they maintain consistent internal proportions. This arrangement might be advantageous in them being able to efficiently adapt the lengths of their toes independently of one another, but still maintaining the internal proportions that are necessary to create a grasping morphology. There's a similar lack of covariation in the quail, which is closer to the hypothesis that we had of greater modularity in a non-grasper, but with a lot more variation in the proportions of the hind toe, which perhaps indicates the less important role of maintaining consistent proportions in the hind toe in a species that doesn't use their foot to grab things. Conversely, the covariation between toes is much more significant in the zygodactyl birds down the bottom, which follows our prediction for grasping birds. Interestingly, their internal proportions also vary much more. So it's possible that this reflects a greater need to balance the opposing toes in birds that use their feet for tasks such as clinging to vertical surfaces or manipulating objects rather than simply perching. My final avenue for inquiry in this part of my project is to assess whether these differences and similarities in integration and modularity impact the capacity of foot morphology to evolve. My findings are extremely preliminary, but the values seem to suggest there are only slight differences in how each foot morphology may be able to adapt to environmental change. From initial investigations using quantitative genetics methodology, which seeks to predict how heritable traits could vary in response to selective pressures, it seems that the rosella might have a slightly greater capacity to evolve in either direction along that trade-off in foot proportion, and possibly at a greater rate. This would be interesting given how much more of the morphospace is occupied by non-zygodactyl birds. However, the difference is negligible using other statistical measures, such as a random skewards method, so I aim to further explore this area moving forward. To conclude, the impact of specialization on the range of forms that might be available to an organism is a topic with some fascinating areas to explore. My goal for my research is to be able to build on the concepts of biological trade-offs and constraints to identify how we've come to witness the wide range of avian forms that exist today. Mainly, I seek to extend this analysis further across the phylogeny to see how other foot shapes might be responding in this context. However, I hope this work will also be useful in helping us to speculate on how the specialized forms of birds and mammals might help or hinder them in adapting to future changes in their environment. As I stated, much of the analysis of my data is ongoing, so I'm happy to receive feedback from any audience members who are interested in discussing the approaches that I've taken in my research so far. Many thanks to everybody for listening, and thank you to my supervisors, my friends, my collaborators, and the institutions that have made my research possible. Thank you.
Excellent.
A quick one.
Do you have questions for Ellen? Elizabeth? Oh, is that just clapping? So I have a question, Ellen. So how much of the relationships between toes do you think in what you've looked at so far is based on developmental constraints, or is it really that you get integration because of natural selection features on it?
Well, it seems that the case is now, since those values are so close, despite all the different ways that they use their feet, that it is likely to be a lot more of a developmental thing, which was one of the reasons I wanted to put those two different zygodactyl birds in, because they have different developmental pathways that led to those foot forms. And also, there's an extremely different developmental pathway between the quail and the perching bird in all of these toe reversions that they've gone through in their evolution. So it's looking that even though they've done all of that, it really is a developmental constraint, maybe more than it is an environmental one or a selective pressure. At least that's what I'm thinking at the minute.
Right, good. Brendan, you have a question?
So either Mark or Janina, you're going to need to ask them to unmute.
Oh, right. Unmute.
Now. Can you hear me?
Yes.
Okay, perfect. Thank you for the talk. It's very interesting. I was wondering if there's a potential, or if you've considered using fossils to look at maybe an even more ancestral form of the bird foot. I know you said that the quail kind of represented that sort of ancestral form, but I'm wondering if you could go even further back by looking at fossils of the earliest birds and bird ancestors.
Yeah, that would be really cool. Especially one of the groups of, old extinct birds that I'm particularly interested in are early perching birds that still have a zygodactyl foot form, because one of the hypotheses was, why didn't those guys last longer than they did when the rest of the perchers did so well? And is it because that foot form is so different? It would be really cool. Unfortunately, I would need a really big sample size for that kind of thing to do any amount of evolvability work on it. But finding where they land on that sliding scale, it's really a cool way of figuring out what type of ecology that bird might have had in the past. So yeah, fossils definitely do come into play in a lot of this type of morphological study, but maybe not so much more for the evolvability stuff. Thanks.
Right. There's also a question in the chat. Someone asked, is it possible to incorporate uncertainty in the inference of ancestral states into these models?
I'm not so sure in terms of the quantitative genetic stuff. It's not something that I'm super well read on. Definitely, you could in terms of the morphometrics that I'm more accustomed to, you can use that for inferring a lot of ancestral state type stuff. But yeah. In terms of the second part, I'm not as certain.
There's also another question in the chat. How was evolvability measured?
So that is part of the quantitative genetics approach, that it takes these covariation tables and sort of it introduces a bunch of different selection gradients to that data, as I understand it, and then tests how well that the mean of that population can move in certain directions, and whether it's able to slide along in against different or the same selective pressures, and how easily those values can be moved in that space, if that makes sense. It's a theoretical approach to it, more so than actually, breeding a whole bunch of quails and then measuring it that way. Easy.
Excellent. We've got about a minute till the next talk. So the next talk is by Abhiski Gopal, and it's a recorded talk, but I think Abhiski is here if there are questions. So
Yes, I can just share the talk. Okay. So let me know when you want me to start the talk.
You can start it now. It's 9:15.
Okay. Okay.
Hello, all. Thank you all. Center for Cellular and Molecular Biology. And we are working on select woody plants of Western Ghats and Peninsula, where we're looking at the biogeography and diversification history. And today I'll be talking about one such group that we're working on, that's Melusa. So there is disparity in how clades, biomes, and forest types are distributed across the globe. And one way to examine these is through the lens of historical biogeography, which primarily deals with explaining the present and the past distribution pattern of species and the underlying environmental historical drivers. And it delves on questions like, why are organisms distributed where they are today? One such way to examine this is through the lens of historical biogeography, where the spatial information is explicitly integrated using the temporal information using dated phylogenetic trees. And 1 second, let me just got it. And which is illustrated here that given a clade and with this extant distribution shown here and on the tips of this phylogeny, their most likely ancestral ranges are estimated in a likelihood framework. Now, the colors in the pie chart correspond to the probability of a given area at that particular node. Now, South and Southeast Asia, one of the most specious regions in the world, having high diversity and endemism. It's also one of the most geologically dynamic regions in the world, where both ancient Vicarian events and more recent sea level fluctuation in the Pleistocene have shaped the distribution patterns of the extant taxa. So for the purpose of my talk, I'll be focusing on this region with a particular focus on the peninsular India. Now, coming to the Indian context, some key geological events are highlighted that India split from Madagascar around 18 million years and was an insular landmass drifting northwards. Extensive volcanic activity towards the end of the Cretaceous triggered mass extinction in the peninsular India, affecting northern Western Ghats and parts of central Western Ghats more, while southern Western Ghats were relatively stable. Sorry. And furthermore, the aridification of the peninsula India, along with the intensification of the monsoon, led to east to west contraction of the wet forests, which are currently now restricted to the western slopes of the Western Ghats and also pockets of high elevations in the Eastern Ghats. We explore the influence of these past geoclimatic and biogeographic processes on the extant distribution using Melusa as a case study. It is part of Ananaceae, which is a large pantropical family with around 2,000 species, and it is one of the most diverse family within magnoliates, which is one of the basal clades of angiosperm. 4 major hypotheses have been put forward to explain the origin and distribution of Ananaceae in Southeast Asia. However, recent studies have found support for the Miocene geodispersal hypothesis, where exchange of taxa occurred over the Arabian Peninsula during the warm phase in Miocene. And there has also been evidence of long-distance dispersal from Africa to Southeast Asia and vice versa. So Ananaceae, while pantropical, are restricted to primarily wetter forests, implying strong niche conservatism. While at the coastal scale, both at the broad geographic scale and at the taxonomic level, that is above the genera level, the biogeographic processes shaping the distribution of Ananaceae are relatively well understood. But what we are currently lacking is more fine-scaled understanding, both in terms of the geographic scale and also the taxonomic resolution. And this is especially more in the case of Southeast Asia, where due to limited sampling and lack of phylogenetic data, there is limited understanding in terms of these processes which have shaped the current distribution patterns. So we explore this gap by examining Melusa, which is a tropical genus having around 60 species restricted to South and Southeast Asia. Previous studies from Southeast Asia have recovered 4 major clades within Melusa, which can be broadly characterized by their key floral traits. However, there are no representation from the peninsular India. So peninsular India after Indo-Burma is one of the richest sites for Melusa, having 15 species, out of which 13 are endemic. Almost all of these are range restricted distribution within the biogeographic subdivision of Western Ghats. For example, you have 5 species which are restricted to below the Shenkota gap. You have 5 species which are restricted to Nilgiris, and you have some species which are found in the Eastern Ghats. Apart from these 13, you have 2 non-endemic lineages, Melusa tomentosa and Melusa velutina, which are more widespread and found throughout the peninsula. So given this, our objective was to reconstruct a time-calibrated phylogeny of Melusa with the inclusion of the endemic Indian Melusa, to examine the role of dispersal and climatic variance on the current distribution patterns, and to examine the patterns of lineage accumulation across space and time. So phylogenetic reconstruction was done using a likelihood and a Bayesian framework with 5 plastered markers. Overall, there are 52 species in the phylogeny, with 15 species occurring in India, representing complete sampling of the Indian Melusa. Molecular dating was done using a Bayesian approach, and appropriate constraints were given following previous study. The dataset was run using an uncorrelated relaxed log model, assuming a long normal distribution of race, and it was run for around 5 million generations. Ancestral ranges were estimated using BioGBS, which provides a likelihood framework that models how branches in a phylogeny have shifted among a set of discrete ranges over time. Now, these shifts in ranges can occur along a branch, which are called as anagenetic events, which includes processes such as dispersal, extinction, or they can occur at the time of branching. And these cladogenetic events can be either sympatric, vicariant, or founder-event speciation. 7 areas were defined based on the extant distribution and biogeographic zones. An unconstrained model was run with maximum allowed areas to be 3 at any time point. The best model was selected based on AIC scores, and to account for uncertainty in the ancestral range estimation and to summarize key biogeographical events, 100 biogeographic stochastic maps were run on the best model. Now coming to the results. Our key results shown as a Bayesian tree with the probability values indicated above the nodes, and stars indicate the species for which we have added the molecular data. So our key results is that Melusa is monophyletic. Similar clades as previously reported were recovered with strong support. We recovered 2 new clades, that's Melusa nilgirica and another clade F, which is represented by primarily the Indian endemic clades and also some species from Sri Lanka. And these clades broadly have a similar morphology as what has been reported for floral morphology, as has been reported for clade P. And coming to the dating, the divergence in the estimation show that Melusa is likely to have originated in mid-Miocene. The Melusa nilgirica is likely to have diverged around 11 million years from the Indo-Burma and the Peninsular Indian clade. The crown age of Peninsular Indian clade is around 9 million years. And overall, most of the divergence in this particular clade have occurred in the late Miocene, post 10 million years. Now, the biogeographic results overall showed similar results across all the models run. However, DEC plus J had the least AIC values. So here, as mentioned earlier, the geographic distribution of the extant taxa is indicated at the tips, and the ancestral range is estimated. The colors in the pie chart correspond to the probability of area occupied by the particular node. Now, the results show that the ancestral area of Melusa is likely to be Peninsular India and Indo-Burma, which post sympatric speciation resulted in a clade with a Southeast Asian distribution and a clade with Peninsular India and an Indo-Burman distribution. The Peninsular Indian clade is likely to have originated via climatic vicariance around 10 million years, which is associated with the monsoon intensification and the contraction of the wet forest. And furthermore, we found evidence for dispersal from Western Ghats to Eastern Ghats at 4 million years, and a jump dispersal from Western Ghats to Sri Lanka at 7, and from Sri Lanka to Eastern Ghats at 4 million years. Also, we find an evidence for a long distance dispersal from Indo-Burma to Sulawesi. And shown are the results of the stochastic mapping, which overall show that narrow sympatry and anagenetic dispersal are the key biogeographical process shaping Melusa, divergence and distribution, and which together account for almost 80% of the total events. And our results of the stochastic map shows that the Indo-Burma has likely played a key role in dispersal to these other regions. for Melusa as a clade as a whole, while within the peninsula, the Western Ghats are likely to have played a key role in dispersal to the adjacent regions. Now shown is a tempo of lineage accumulation in different biogeographic regions, with the regions within the peninsula clubbed into one called as A, with the results show that overall, that both the Indo-Burman and the Indian clades have started diversifying much more earlier, and subsequently when diversified much more earlier with rapid accumulation and eventual saturation of the species, whereas the clades found in Sundaland and in Papua New Guinea, Australia have started diversifying much more recently. We investigated this much more specifically with using more sophisticated models of common and cladders, and the results show that for both Melusa as a clade as a whole and the Indian subclade, both show drastic slowdowns. And slowdowns can result from either density dependence or specialization, but these are generally hard to infer and need to be inferred from multiple angles. So now to summarize, our results show that Melusa is a relatively young clade having diversified rapidly in the late Miocene and Pliocene. Sympatry and dispersal are the key biogeographical process for the clade as a whole, with Indo-Burma playing a key role in dispersal across the regions, which is also shown for several other taxa where the more stable Indo-Burman region have allowed for higher dispersal, often asymmetric to the adjoining areas. Within the peninsular India, the Western Ghats have played a key role in dispersal to other regions. The Indian clade is likely to have originated due to climatic variance, with subsequent dispersal shaping the extant distribution of species. Overall, and the Indian subclade of Melusa shows slowdowns which needs to be investigated further. Now,
Excellent. We have a few minutes for questions. If anyone has a question. Oh, clap. Gustavo, do you have a question? Unmute, please.
All right, can you hear me?
Yes.
Okay, perfect. I do have a question. You said you used two calibrations for the analysis, for time calibrated analysis. So one with a log normal distribution and one with a I don't remember now, but I think it's from the root age, I believe. My question is, how is the fossil record, and do you plan to include more calibrations for the analysis? Do you think this would help with estimating the divergence ages, or it would influence in the future?
hey, thanks for the question. Am I audible?
Yes.
Yeah. Yeah, that's a valid point. But I think in generally for Ananesi as a clade for whole, these two fossils have been well recognized as to in terms of where their position can be. And generally, because these are primarily restricted to within the tropical realm, there have not been many fossil records which can be reliably placed with enough morphological evidence as to where they go. So currently we are still contemplating how best, but I think for now, we're just going to be restricted to these more deeper level fossils in terms of the dating for the clade. But yeah, definitely having more fossils which are closer to the clade of interest would definitely help in better divergence estimation. But I don't think currently we are there yet.
Good question. Anyone else have a quick one? No? Excellent. Thank you, Abisky. Our next speaker can get their screen sharing going. Our next speaker is Joe Baker, and we've still got a minute or so. So, Joe, why don't you get your screen share up and then hold off for just a second if anyone else is going to come in. Excellent. And Joe, are you unmuted? Yes, good.
Yeah, should be. Can you hear me?
And you can hear us now too, so.
I can still hear you, yeah.
Excellent.
I can no longer see you though, because I am on a laptop, so just let me know when I'm ready to go.
Okay. just give it another minute in case someone else is coming in.
Sure. I'll just take the opportunity to say good afternoon from the sunny UK. Uncharacteristically warm.
Yes, we've been in the 90s in New England, so uncharacteristically warm here as well. So why don't you begin?
Yeah, sure. So I'm Jo Baker. I'm a postdoc here in the University of Reading in the UK. And part of the last couple of years, I've been working on a project looking at hominin evolution. And one of the focuses we've been zooming in on is to look at the coevolution of encephalization and manual dexterity in hominins, but also other primates. So I'll start off with just a bit of background to the topic. As probably many of you already know, humans, our own species, we're quite obsessed with our own unique characteristics. And one of the ones that comes out the head of lots of headlines and lots of beginnings of every paper is that, oh, humans are very intelligent. We have very large brains relative to our body size. That's true across the animal kingdom, but also compared to other primates. So here on this graph is a plot of brain mass against body mass for primate species, and the blue dot indicates the human. So this idea that we've got very big brains relative to our body size kind of leads the way in conversations about our intelligence and our cognition. It's not surprising that humans are argued to be one of the most intelligent animals on the planet. I say one of the most because, you know, there are other humans around that perhaps aren't fitting the bill like the average human. But in general, human cognition is also linked to dexterity, and this is shown in experimental studies in individuals rather than at a species level, but actual lab experiments that link dexterity to cognitive function, so things like memory and information processing, they are strongly linked within our own species. This idea that dexterity and cognition are linked is often highlighted and brought up over and over again in the discussion of tool use in our own species and also our hominin ancestors. So when exactly the origin of tools was in the fossil record is very contentious. There's a lot of debate about which species were the first to use tools and when and where it happened. But what is commonly highlighted is our cognition, our actual ability to understand the need to use those tools in the first place. However, hominids are not the only primates to use tools. There's lots of examples throughout the primate phylogeny of species that have been demonstrated on at least one occasion, but often on many occasions, to not only source tools, but also manipulate them for their own benefit and their own purpose as well. In addition to that, tool use is just one example of the wider suite of behaviours that is observed across the animal kingdom, known as extractive foraging. This idea of our interactions and manipulation of our environment in order to gain some sort of benefit, in this case usually food, is widespread. It's observed even in invertebrates. This link between behavioural complexity and brain size could still exist, and there is some evidence that behavioural complexity, or measures of manual dexterity in terms of behavior, is also linked to brain size. But how exactly can this be, how exactly has this manifested in the evolution of our hand and in the hand of primates? When we think about how widespread these behaviors are, what morphology do we have that is specifically associated with the kind of dexterity that we see within primates? So focusing in again on humans, lots of traits have been linked to manual dexterity, and this slide is deliberately busy just to show you that there is a huge amount of traits just within the single organ of our hand that have been linked to our ability to have highly refined precision grips. And I'm not going to talk about all of them, we're just going to focus in on one of them that is very easy to measure, not only in our own species, but in other living primates, but also in the fossil record as well. And that is a long thumb relative to finger length. Thumb length is remarkable in humans and is often highlighted as one of the characters that makes us what we are. So thumb length is highlighted as uniquely human. A modern human thumb length, shown by the green bar here, relative to the fourth digit here, is much, much longer than observed in our closest relatives, the other apes. And while there are some primate species that do overlap, these tend to be species that also show high manipulation complexity, leading to the strong suggestion that longer thumbs lead to higher manipulative abilities, and thus you would expect to see that species with long thumbs are more likely to use tools or show complex manipulative behaviors. However, this is all stemming from the fact that long thumbs facilitate opposability. So the idea that humans can have a very precise grasp is linked to our ability to have pad-to-pad precision grasping and opposable thumbs. But we know that opposability isn't just limited to humans, and not only that, but we also see really highly precise grasping in behaviors, even with only pseudo opposability. So this is a capuchin monkey that shows really refined grasping behavior, the ability to manipulate really tiny objects between its first finger and its thumb, but it doesn't have pad-to-pad precision. It only has pad-to-side precision. So this is an indication that thumb length might just be a more relative measure of dexterity in general rather than tool use. And this leads us to our expectations between brain size and thumb length itself. So our first question is, do species that use tools have longer thumbs? We know that there is a high number of primate species in the living species that we have today that have been demonstrated to use tools in different forms. Do they have longer thumbs? If we think that longer thumbs are more indicative of an ability to manipulate. Not only that, but we also expect a positive association between thumb length and brain size. If we think that thumb length is a proxy for dexterity and brain size is a proxy for cognitive processing ability, then we expect these to have co-evolved across all primates. Of course, if we don't see any relation across all primates, this might mean that thumb length isn't a good measure of dexterity at all, and perhaps it's associated with other things that are exerting selection pressure on hand morphology, such as locomotion style. These are just 3 species that have highly divergent locomotion, which are likely to have exerted different selection pressures on hand morphology. And we also have to remember that thumb length in isolation might not be enough to link to brain size, and instead that these different features that we think are associated with dexterity might be acting as a suite of traits to improve precision grasping. If we think that humans are unique, this association could be entirely driven by hominins. That is, that we would see an increase in relative thumb length that is not observed in other primates, and that would lead to an erroneous observation of thumb length and brain size across all species that is purely driven as an intercept difference between non-hominins and hominins. Of course, we could also observe the overall relationship and some intercept differences in hominins, but in this case, because we observe a relationship between dexterity and cognition, it implies that any outliers are being driven by selection on something other than manipulative ability. Or we could find that hominins conform to the primate pattern, and that is that both relative brain size and thumb length might seem unique in humans, but in actual fact, they represent the ends of a continuum. They are just the extremes of a continuous variation of morphology that are part of an underlying single uniform process that are acting across all primates. Now, this figure is going to be hated by some people that are familiar with brains, but it's just a kind of diagrammatic figure to represent the fact that different brain regions have different expectations and different functions regarding the types of behavior that they are implicated in. And we do expect that the brain regions guiding visuomotor control, so the brain regions important in vision and in motor control and management of complex behavioral sequences, to both be important for manual dexterity. And so in order to do this, we have, in order to account for this, we derive another set of expectations in that we expect both the neocortex and the cerebellum, which we expect to act as a sort of unit guiding visual and motor control, to both be important. In that case, we would expect both neocortex and the cerebellum to increase in size along with thumb length. So what do we do to test our expectations? We collected thumb length, finger length, and brain mass for 95 taxa across the primate tree of life, and this includes both extant and extinct species. The green bars represent thumb length, and they are superimposed on top of fingers, which are the second metacarpal here in blue. We have brain size data for all species in the tree, and they are indicated by the red circles at the tip. And then we also collected neocortex and cerebellum volume for a subset of these species. Obviously, this is only available for extant taxa, so there's only a limited portion of these available. And in hominins, we obviously only have the human, so any inferences about hominins will have to be made from our own species in this case. We test our expectations using phylogenetic generalized least squares models and a phylogenetic predictive modeling approach to identify whether hominins are outliers to any of the predicted models that we infer. So the first question, does tool use predict thumb length? And I'm really sorry, but the next slide is going to be a bit stark because I only just did these analyses today. And the answer is no. So actually, there is no association between thumb length and tool use of any type. And we tested lots of different things. So there's a huge compendium of tool use data, and that includes tool use, true tool use, so animals that actually seek out a tool for an intended purpose, tool manufacture, so animals that modify, primates that modify tools for use, but also we excluded records that have only been observed in captivity and records that have only been observed in a single individual, and nothing made any difference. There is absolutely no association between thumb length and tool use across all primates. How about the relationship between thumb length and brain size? Well, first we find that thumb length and finger length are strongly linked. And when you add brain size into the model, that is retained. But we also find that brain size is a very strong predictor of thumb length. This is in line with our expectations, but when we remove hominins from this analysis, it makes absolutely no difference whatsoever. So this is an indication that humans are not, or hominins are not driving this pattern. And in fact, that this is a primate-wide association, the relationship between thumb length and brain size. We then also find a similar association with the neocortex. We find that neocortex volume is strongly associated with thumb length, but actually there is no association with the cerebellum. And this is interesting because we expected both of these to evolve as a kind of unit, but it's an indication that seems to be upheld no matter what we do. So if we test it in subgroups of the tree, if we remove species that have been identified to have interesting proportions of neocortex and cerebellum, all these relationships are still upheld. So there is something quirky going on in the neocortex that is associated somehow with manual dexterity. As I said, these results are not being affected by the inclusion of hominins, but what we wanted to do is test to see whether hominins actually fit the expectations of these models, or whether or not their thumb lengths are identified as outliers, despite being part of the primate-wide association.
That's 12 minutes.
Thank you. So hominins do have significantly longer thumbs. So hominins have much longer thumbs than is expected for other primates, but they are no longer outliers when we account for brain size. And this is an indication that brain size is explaining the long thumbs that we observe in our own species. So large thumbs, large brains and long thumbs aren't uniquely human. They're just the ends of a continuum that is a process acting across all primate species. And this is also true for the neocortex. So what do our results mean for tool use in hominins? The short answer is nothing. So we can't really say anything without any more information on earlier hominins or some sort of index about whether the length of, what length of thumb indicates more likelihood of tool use without more information. So I won't dwell on this, but I will just point out that Australopithecus sediba is the one exception to our results. And Australopithecus sediba shows hands that has been recently demonstrated to have adaptations associated with multiple styles of locomotion as well as manual dexterity, which fits our idea of the fact that outliers might be identified in the case where species still have high manual dexterity, but also an alternative style of locomotion. So this is the brief structure of a paper we're currently writing called The Coevolution of Encephalization and Manual Dexterity in Hominins and Other Primates, co-authored by Rob Barton and Chris Venditti. And I'm happy to take any questions if there's time. Thank you.
Excellent. So we have time for one very quick question.
Sorry about that.
No problem. Any? Oh. No, we should probably move on to Andrew. Thank you, Joe.
Thank you very
Andrew,
much.
do you want to share your screen?
Yes, yes. Can you hear me?
Yes.
Okay.
Give it just one minute.
Sure. You can see my presentation now.
Yes, very good. There we go. And Andrew, please introduce yourself and begin.
So, hi everyone. I'm Andrew Helmstetter, and I'm a postdoc currently working at the University of Montpellier. And today I'm going to talk to you about some of our work where we characterize the reproductive strategies of flowering plants, or at least try. So angiosperms, or flowering plants, are a particularly large and successful group. They contain about 300,000 extant species, and depending on who you ask, have evolved over a period of about 270 million years. During this time, an exceptional diversity of reproductive traits have evolved, including different modes of pollination, different floral traits, different fruit morphologies, different ways that seeds are dispersed. Not only this, but also there exists a wide range of reproductive systems. For example, from obligate selfing species to outcrossing species, from hermaphroditic species to dioecious species with separate sexes. And actually in nature, most angiosperms are outcrossing hermaphrodites. We've often been interested in how this diversity of reproductive traits is associated with differences in species richness among different angiosperm clades. Our review in 2023 showed that these reproductive traits are actually the most commonly investigated type of trait in studies of trait-dependent diversification, where you want to link a trait with differences in diversification rate. However, actually, when you look at these studies, most of the time, these traits are tested in isolation, so on one at a time on their own. Yet, it's important to remember that traits don't exist in isolation. I made this slide, I feel like it's almost a motivational poster for the poor traits we find in nature. But it's important to remember that when we look at species, we can see that they're actually combinations of traits that are the result of evolutionary processes like natural selection that themselves are embedded in ecology, the environment, and geography. And what we would like to do is to make this additional variation visible. And one way we can do this is by building a multivariate trait space. So, for example, here is a functional trait space that was based on more than 40,000 seed plant species from using 6 classical vegetative plant traits from Diaz et al. In 2016. And the main axis of variation in this space was related to woodiness, with the woody species on the right and the herbaceous species on the left. But other traits, such as seed mass or the height of plants, also contributed to this axis. So while we know quite a bit about vegetative trait associations, we know relatively little about associations in the reproductive trait space. We've had some long-standing ideas about simple trait associations, such as selfing and short lifespan. We've had these for some time, and some recent studies have gone beyond this to construct trait spaces using reproductive traits based on plant pollinator networks or traits from regional, in this case, European flora. And both of these studies demonstrated that reproductive trait variation adds important information beyond these classical vegetative traits if we're examining the angiosperm functional trait space. However, these studies themselves actually lack important sources of variation. There are no abiotically pollinated plants here, and there is just one region, a European and Alpine flora here, and no trees or grasses. So the aim of our work was try to encompass as much trait variation as possible across the angiosperm tree of life to answer the question, what are the major reproductive trait associations or strategies at the angiosperm scale? To do this, we needed to build a novel data set that was systematically sampled across angiosperms. So we collated data from 360 species, including representatives from all families with more than 100 species. And if there was a particularly large family, we had multiple representatives. And we assembled a trait data set of 21 traits associated with reproduction, both directly, such as breeding systems, flower morphology, but also indirectly associated traits like growth and life history traits. So using these data, we built a trait space. And here is a plot of the first 2 PCOA axes on our data, where each point is a species. And we see along this diagonal similar to what was found in Diaz et al., this sort of woodiness, size, lifespan axes. In the bottom left, we have woody species that are generally tall. They live long, they have large seeds. In the top right, we have herbaceous species that are short-lived. Perpendicular to this and of equal importance was a reproductive axis, predominantly made up of pollination-related traits. So in the top left here, you have mostly wind-pollinated species that had little floral reward, had unisexual flowers that were dull. And in the bottom right, you had these very showy, biotically pollinated species that had floral rewards and bisexual flowers. And this demonstrated that at the angiosperm scale, these pollination-related traits are making a substantial contribution to this trait space. So as one of our goals was to identify these major reproductive strategies, we performed clustering to try to group species together in our trait space. So first we used a hierarchical clustering approach, and this yielded 3 major groups. One group contained species that we could broadly summarize as herbaceous species with bisexual flowers. A second group we could summarize as woody species with bisexual flowers. And both of these groups were relatively narrow compared to the third group, which contained species with unisexual flowers. And this was quite large, taking up a relatively large portion of the trait space. Though we use these 2 combinations of traits to distinguish our different strategies or clusters, actually behind all of this are a number of different traits that are co-varying, represented in this bar plot here. So although this cluster on the left is summarized by unisexual flowers, actually often these flowers are long-lived, outcrossing, they have long-distance dispersal, they have actinomorphic symmetry, and they have dull flowers. And it's really this ensemble of traits that is defining these different clusters or reproductive strategies. So here is another representation of the same trait space. Actually, this is a UMAP approach. It's a method that can account for non-linear relationships in the data. And these same 3 clusters we identified before are clearly coming out in the trait space. And what we did here was we did some finer scale structuring, finer scale clustering to try to sort of identify substructure between these major strategies. And what we found was that, for example, in the herbaceous bisexual species here in the bottom left, on the left side, where this was actually the structure was determined by floral symmetry. So on the left side, we have actinomorphic species, and on the right side, we tend to have zygomorphic species, where you have a bilateral symmetry. And for the woody bisexual species, the structure within this major strategy was determined by, on one hand, small dull flowers, as opposed to large bright flowers. So what we can do once we sort of defined or described our strategies is we can treat them as compound traits, and we can use them to reconstruct their history on a phylogenetic tree, as this tree here represents a sampling of our 360 species. 360 is an excellent number for plotting a circular tree, not on purpose though. And what we did when we plotted these different strategies as defined by our hierarchical clustering, these 3 major strategies, we found that actually they were highly labile at this angiosperm scale. We found lots of transitions between strategies amongst our different species and across the angiosperm tree of life, but also we didn't find clades of 10 or more species that had a single strategy. These strategies have evolved and transitioned rapidly throughout the history of flowering plants. One other interesting result was that we corroborated previous work that suggested that the ancestor of angiosperms was a woody plant with bisexual flowers. Now, being evolutionary biologists, we were also interested in how phylogeny might affect the trait space. So what we did is we took all of our traits and we fit, depending on whether our trait was quantitative or qualitative, Ornstein-Uhlenbeck or Markov models to each trait. And this gave us a set of model parameters that we could use alongside our tree to simulate data sets. And then we took these simulated data sets and we ran them through the same pipeline to generate trait spaces. And then we took these trait spaces and we compared them the simulated ones with our empirical trait spaces. So this plot on the right here, you have on the y-axis the relative eigenvalues, so the explanatory power of the PCOA axes in the trait space, and you have your PCOA axes starting with the most important on the left. And actually, what we found was if we take these two colored lines here, our empirical data, the amount of variation the axes explain in the first, the most important axes, the empirical data explained a lot more of the variation than our simulated data, these error bars in black here. But as you went down to less and less important PCOA axes towards 4 or 5, they began to overlap, which suggests that perhaps once you get down to the 4th or 5th most important axis, this might be just an effect of phylogeny, and the interpretation of these axes should be taken with a grain of salt. We also looked to see whether in our simulated data any traits came out as the most important, and we actually found that wittyness was often very important, and we attributed this to the fact that in our data set we have a roughly 50-50 split of witty and herbaceous species. So if you're going to structure a data set and you can split it in half, it would make sense to use that trait. So just to summarize, reproductive traits, they're critical to the evolutionary and ecological success of species, yet they're not so often used in studies of functional ecology. And we have added to growing evidence that they should be taken into account because they provide important variation that gives a new perspective in plant functional ecology. We identified 3 major strategies, sort of broad angiosperm scale strategies that were based on wittyness and flower sex, amongst various other traits, but also substantial substructure within some of these strategies. One surprising outcome that we weren't necessarily expecting was the different contributions of sexual and mating systems. So while mating system was broadly correlated with the size, wittyness, lifespan axis, actually sexual system was really important in structuring the trait space. This indicates that these sort of different avenues towards achieving outcrossing can have different effects on the reproductive strategies of flowering plants. In the future, we're interested in trying to link the reproductive strategies that we identify with species diversification. However, as you can see, given our sampling, this might be intractable at a broad angiosperm scale. So we're interested in comparing different reproductive strategies across families. Are the reproductive strategies in Solanaceae the same as those in palms or Ananaceae? And then once we've defined the reproductive strategies, can we link them to patterns of diversification? And one of the sort of pipe dreams behind this is that many modern models of trait-dependent diversification include hidden states. So these are unknown traits that can influence diversification alongside your trait of interest. If we use these kind of multi-trait approaches, such as defining broad-scale strategies that encompass lots of different aspects of species biology and clades biology, can we unlock the secrets behind these sort of unknown black box hidden states? So with that, I'd like to thank everyone who made this work possible, particularly Sylvain and Jos and the rest of the diverse working group, and the funders for providing the funds to do the work. If you're interested in learning more, we have a preprint out that goes into more detail. And thank you for listening. And I'd be happy to take any questions.
Excellent. Excellent. Do we have any questions for Andrew? Yes, Neil. Unmute. Neil, did you have a question?
Suspenses.
Yes, now you are unmuted. Now,
now you're muted
again.
There you go. No,
okay.
Stay there. Neil.
You do it, Neil. I believe in you.
Episic.
Neil, you could also just type it and we can read out a question.
Yeah. Episic, do you have a question?
Yeah. Hey, thanks, Andrew. Great talk. Just thinking about extensions of this, and if you were to look, because I think from a community ecology point, there has been a lot of work on trait syndromes, right?
Mm-hmm.
And so just looking at, if you look at taking a step back, if you also look at conservative or accusative strategies themselves, And if you map that on a phylogeny, how does that look? And what's your take on that?
Yeah, a lot of the work behind this was started with syndromes. We knew there were a lot of syndromes, but our sort of interpretation was that the fact we didn't see, for example, the selfing syndrome didn't really come out in our broad scale analyses. And we thought that perhaps what that might be is because of the context in which these syndromes evolved, you don't capture that at the huge variation at the angiosperm scale. So that's kind of the idea of going into the family level analyses is, do we see some of those syndromes that are, have been identified, pollinator syndromes in particular groups or in particular communities when you zoom in, and how do they compare to what we see at a broader scale?
Got it. Got it.
Great. For everyone, that's the end of this session. We can keep it open if people would like to talk to the presenters for a little bit. But if not, have a good day.
I just see Neil's question in the chat.
Oh, there we go. Yes.
He says, you mentioned that there's lots of transitions between the three clusters. Are there any transitions that aren't seen, e.g. strategy A to strategy B never happens? I can't remember off the top of my head. And we sort of avoided interpreting the transition rates because of our sampling. But I remember that transitions between each strategy were common. There wasn't a sort of a one-way transition matrix.
And there was one other question in the chat, I think. Did different traits fit different models of trait evolution?
So
it's a little, it's right above Neil's.
Yes. So we had a relatively simple approach to testing models of trait evolution. We wanted to compare rates, so we actually just used an equal rates model when we looked at the rates of transition of all of our different traits in our data set, because we wanted to have one rate to see, does one trait diversify faster than another trait. Again, because of our sampling, we didn't really have much confidence in the rates given how many species are missing in our phylogeny. But there's definitely an interesting question. And once again, applying this at a family level, you can really start getting into that because you're starting to have a higher sampling fraction, and you can actually more reliably do model testing of your models of trait evolution.
Excellent. Any questions for any of the other presenters? No? Well,